Okay, folks, so just doing a quick vid to try and clarify a couple of things with our mass spec. So here's the example that I was showing you in class, where we're actually showing that the parental peak has a mass of 59. You'll notice on this there's actually a very small bar on 60. And this is an error in this question or in this spectra because the parental ion should have been labelled 60, but in this case they've labelled that one 59, which is not the parental ion. It still might be a fragment that's produced, but it would be the parental ion that's lost to hydrogen. So it would have a molecular mass that's one less than the parental ion. So let's go to the correct one, and as you can see here, this one's labelled a little bit better. So lesson learnt, do a bit more research, don't always go with the first thing you see. But here quite clearly it shows us that the molecular ion or the parental ion or the parental molecular ion even, lots of interchangeable terms for that, remember, has a mass charge ratio of 60. Okay, so for us, what does that mean? It means the molecular mass of the parent molecule is 60. So then from that, we would establish some possible combinations based on the information we're told in the question. So the question I showed you told us that we had carbon and hydrogen and oxygen that made up our molecule. So you just trial and error and experiment with different combinations of those until you got one that fitted. Hopefully that was fairly clear in class with the work we did on that. But the other thing worth just mentioning here is the base ion and how that's different to the molecular or parent ion. So the base ion, remember, is the most abundant. So it has the highest peak. And you'll notice here as well the scale is actually correct. So that other one that I showed you was also correct with the scale because the abundance of that base ion is always going to be 100%, and everything else down below that will be relative to that. So here it actually labels that too as relative abundance. So I think that's important. Some of the spectra that you'll see will be simplified spectra, so you may not get every single peak. And here, the other thing I was going to point out, I don't know if it's that easy to see, but there is a very, very tiny bar on this point here. At 60, it just doesn't show up very good when I was doing this imaging, but there's a tiny little bar there. So on the exam, just want to clarify with you, it would be very, very clear what you need to know and what the peaks are and what they're labelled. So hopefully that just reassures you of that. If we look down at our lowest peak here, I will just do maybe one example. Went through this in class as well, but just to clarify, so looking at the lowest peak, using the scale, we can see it has a mass of 15. So if it has a molecular mass of 15, and you know that the molecule is made up of carbon, hydrogen and oxygen, basically we rule out oxygen because oxygen has a mass of 16, so it can't be oxygen. So therefore it has to be a combination of carbon and hydrogen. And in that case, we would have 1 carbon and 3 hydrogens. So this would be a CH3 group down here with a molecular mass of 15. Hopefully that sort of makes sense, folks. And it clarifies any confusion that resulted from yesterday's incorrect example. I'll do a second part in just a second. So just bear with me for 2 ticks. Okay, folks, just wanted to talk a little bit to you about aldehydes and ketones and give you another mass spec example using a ketone. So we talked about these type of bonds a little bit in class, but basically a ketone is when you have a carbon bonded to 2 other carbons that are not shown here in this part of the diagram, and it's double bonded to an oxygen. Okay, very, very similar functional group is an aldehyde. So the carbon is still double bonded to an oxygen. One other bond of this carbon would go to an adjacent carbon, but the other one goes to a hydrogen. So that would be the end of the molecule. whereas this would be part of the middle of the molecule. So that's the key difference on those. I'll flick you this website as well, which sort of goes through functional groups nicely with some examples. So I'll give that to you through our various forums for sharing files. The last thing I just wanted to spend a few minutes on is an example of a ketone. So this sort of leads into the work we're going to be doing on naming molecules as well. So in this example, you're given a simplified mass spectrum, so it doesn't show all the peaks necessarily that might be produced in a mass spectrum, but it shows the ones that you need to consider. And it gives you the name and also the semi-structural formula. So remember, the semi-structural formula is just how you would draw this molecule. So you'd have a CH3 on the end, bonded to a CH2. Here's our ketone here. So we've got carbon on one side. Oops, that didn't work so well. So let me just get out of that. I was trying to highlight, but that didn't work so well. So here's our ketone group here, okay, with the oxygen and the carbon. Here's Tilly. Hi, Tilly. Say hi. What did you just give me?
Popcorn.
Popcorn. Did you make some popcorn? Thank you. Do you want to say hi? Say hi, everyone. No? Okay. Thanks for the popcorn. Okay, so that's our ketone group there. So this carbon would be double bonded to the oxygen, single bonded to this carbon over here, and single bonded to this carbon over here. So hopefully that's pretty clear with that. And the name, remember folks, we're looking for the longest carbon chain. We've got 5 carbons here in a straight linear chain, so it's going to be a pent group. The position of the actual ketone group is on the 3rd carbon, so hence the 3, and the suffix or the ending of the name is -one. And that comes from the actual name of the functional group. So this molecule, here's our parental ionic peak, and that means this molecule would have a mass of 86. But just remember, folks, that it would have to have a positive charge here. So for it to be detected on the spectra, it needs to have a positive charge. So if you were asked that question, you would actually lose marks by forgetting to put that positive charge on. So just remember, folks, these are all positively charged molecules that are being detected. 29 is one that you'll probably see a bit in a lot of the examples we look at, because if we look at this part of the structure here, the CH3-CH2 group has a molecular mass of 29. So if you're asked to identify that part or that fragment for this question, it's come from the CH3-CH2 group. And so to get full marks for that on your exam, you would say that, and don't forget the positive charge to actually get the marks for that. So that peak corresponds to that fragment. What I'd like you to do, folks, is have a go at working out the fragment that corresponds to this peak, which has a molecular mass of 57. So have a go at that, please, before class next. And yeah, I look forward to hopefully having clarified any concerns you had about mass spectra. Shoot me a message if you've still got questions. Thanks, gang. Catch you soon.